The overall objective of gene therapy is to achieve a durable expression of the therapeutic gene, or transgene, at a level adequate to ameliorate or cure disease symptoms. Monogenic diseases are disorders caused by a mutation in a single gene and thus are thought to be ideal candidates for gene therapy. To deliver a gene into a cell, a carrier, also known as a vector, is typically used. Can be created by removing the viral genes from a naturally occurring virus and replacing them with the therapeutic gene of interest. Consequently, in contrast to the viruses from which they are derived, the vectors lose their ability to replicate and propagate in the patients. The process by which viral vectors are employed to deliver genes into cells is called transduction. Several types of viruses, including retrovirus, lentivirus, adenovirus, adeno-associated virus, AAV, have been modified for use in gene therapy applications. There are two strategies to gene therapy: ex vivo and in vivo gene delivery. In ex vivo transduction, cells are extracted from the patient and transduced with the gene of interest. These cells are then returned to the patient in a procedure similar to that used in hematopoietic stem cell transplantation, HSCT. Integration is at one or more loci in the patient's chromosomes. In vivo gene delivery involves injecting the vector gene construct directly into the patient, similar to the delivery of other pharmaceutical agents. In vivo gene therapy utilizes a predominantly non-integrating vector, which delivers a gene to a long-lived postmitotic or slowly dividing cell, which, in principle, ensures the expression of that gene for the life of the cell. Frequently, an AAV is used because it is essentially non-pathogenic and has a long-standing safety record in preclinical animal models. To deliver the therapeutic gene into a cell, vector constructs containing the therapeutic gene of interest are generated. To protect the transgene from degradation during transportation to the nucleus, it is housed in the AAV capsid, the protein shell of the vector. Once inside the target cells, the capsid releases the transgene, which acts as a small unit of DNA and functions separately from the chromosome. An episome is typically an extrachromosomal piece of DNA that can persist for a certain amount of time, but that is not integrated into the target cell chromosomes. Transgenes are exogenous DNA sequences that are introduced into the organism of a patient to alter the function of a known gene. A critical element of the transgene is the promoter. A promoter is a regulatory sequence that determines transgene activation. Since transgenes are typically designed to produce a protein, they must have a valid protein coding sequence. This is normally derived from the cDNA for the protein of interest. By targeting the gene that does not work properly and delivering a new working copy, gene therapy aims to provide the instructions the body needs for protein development. With continued progress in gene therapy technology, we are closer to unlocking the promise of gene therapy.